This video covers tests for proportions. So this is for binary data such as survival, yes or no, or if you have a dependent variable that has two classes like male and female. So for this type of data, these tests apply. These are also non-parametric statistics. And we start with contingency tables. So this is just a data preparation step that you have to do. And we'll cover a number of tests that are all good and important. So chi-square test of independence, Fischer's exact tests for proportion, chi-square test of goodness of fit, and then a one sample and two sample z-test for proportions. So these are all slightly different, but they all have their uses and applications. So let's start with contingency tables. This is simply a data preparation step. If you start with a standard data table and you have, let's say, a different species as your class variable, and now you have binary response variables. So this could be a sex, male or female, a survival, yes or no, or you could also code this into ones and zeros, so this is all fine. What you want to convert this to are these types of tables. This is called a contingency table that basically just does a count of all your y's and all your n's in this particular case. So for species A, if this is a longer table, I might have 8 that survived and 12 that died. For species B, 16 that survived and 4 that died, and so on. And you can obviously do this for different variables, so each would get a separate contingency table. And it's pretty easy to do in R. So if you just have a table that contains two columns, so your group variable and your binary response variable, then that's your command here, right? So if that table were to be called dat1, then you just say table of dat1, and this is what you get out. Now in this case, let's say we want to extract the survival data and build a contingency table here. We have to say, okay, we want column 1 and column 3 and build a contingency table from that. Or you could also do it this way if you use your variable names here. Good. So now that we've generated our contingency table, we can test whether the species differ in survival. So this is essentially the same idea as ANOVA. So I have a number of groups. So my predictor variable is a class variable, but my dependent variable is a binary variable. So that's the difference, but the objectives would be exactly the same. So this is my contingency table here that I just copied over. And the first thing that we need to do is calculate all the totals. So 8 plus 12 is 20, 16 plus 4 is 20, and so on and so forth. And this is my overall total. And now I have to create an expected table if there was no effect. So how am I going to do this? I'm just going to look at the columns here, at the totals. So 48 and 32, those are my overall totals across all species. And if there was no effect, they would all have that same ratio. So let's see what that is. 48 divided by 80 is 0.6, and 32 divided by 80, so divided by my total, is 0.4. So on average, 60% survived and 40% died. So now I can fill in my expected values. So I know I'm always expecting 60% to survive, so this would be 20 times 0.6, 12, and 20 times 0.4, that would be 8. And here it's the same, 12 and 8. And for this one here, species C, I have a larger sample size, so 40. And so in that case, my expected values would be just double that, 24 and 16. So these are my observed and my expected values that I have. And now I can do a chi-square test. So the way the chi-square test works is that I sum up my observed minus expected squared over expected. So it's a pretty simple calculation. I start with the first cell here, 8 minus 12 over here, square that and divide by the expected. And then I take the next one here, 16 minus 12 divided by 12, 24, and then I start with a no column. So 12 minus 8 divided by 8, and so on. And so if I sum these all up, I get 6.667, and I can use the p chi-square function just as we had the pnorm and pt function to get my p-value. Degree of freedom is just as in ANOVA 2, so I have 3 treatments level, so t minus 1 would be 2. And of course, you don't have to do this whole calculation here by hand. You can get exactly this result with a chi-square test of that contingency table, of this table here. So if you have this prepared and you feed it to a chi-square test, it would exactly give you that result and the corresponding p-value. And prop test is an alternative function in R that is slightly different, but in this case it does exactly the same job, so you can use either one of those. So just like for ANOVA, you can follow this up with pairwise chi-square tests. In this case, you would feed the reduced contingency table with just species A and species B into the chi-square test or prop test function. And here you actually also have another option, that is the Fischer's exact test for proportions. This is actually a slightly superior test. I would always use this one, not the other two. So Fischer is a smart guy. He actually could calculate the p-value amazingly with combinatorics. So he figured that out. I'm not going to go over this because that is really complicated. But you can check that formula out on Wikipedia. It's just amazing how a human being can come up with something like that. But there's an exact solution to this problem, and that can be expressed in combinatorics. So it's just like the other non-parametric tests that we covered, like the Wilcox rank sum. So these are solvable problems. You can calculate exact p-values. But Fischer's test will not work for more than 2 samples at a time, so you can only compare 2. Otherwise you need the chi-square. In any case, in practice, these are not very different. So only if the sample size gets really small, then Fischer's exact test is preferable. But you might as well always use it. So the chi-square distribution is actually only an approximation. And I should also say that for these types of problems, there are actually more powerful alternatives than chi-square and Fischer's exact test. They are completely different. So before we shift gears to this, I do want to cover another version of this test. And this is the chi-square test of goodness of fit. So that's a slightly different way of thinking about things. And I can illustrate this to you with an example. So an example application for this type of test, an ANOVA situation where you have survival, could be some sort of land reclamation experiment, right? So you planted 3 species and you want to know which ones survive these environments the best. And your question is there a significant difference? Is one of the species better than another? And you know how many trees you planted, so you planted 20, 20, and 40, and that sets you up for that type of test. But there are also other examples where you don't necessarily have both of these columns. So this could, for example, be a situation where you survey. So you don't have a history of what you planted, so you only see one of these columns of what is actually there. Or it could be that the second column is simply hidden. So just to give an example, it could be some sort of fish stocking experiments. Let's say you have a lake and you stock it with 3 species of fish, and let's say we put in 200 of species A, 200 of species B, 400 of species C, and after a year or 2 we come back and we want to see what the survival is. So what I can do is I can sample this lake through casting a net or by maybe fishing for 3 hours or something like that, and see how many of each of those species I catch. So I catch 8, 16, and 24, so just like that here. But I don't know how many died, so I just have a count and perhaps some optional weight variable. So let's say I had stocked them all with equal proportions, 200, 200, and 200. Then I don't need this weight. But if I have stocked them differently in the beginning, or if, let's say, my effort is different, so I might have equal stocking, but perhaps I placed different amounts of effort in catching each of those species. So that's possible that you have those weighting factors. So we can still do our chi-square test with this, but we'll simply do it with one column. And if I have no weighting factor, so my null hypothesis or my random expectation would simply be that they are all the same. So in that case, that would be the total 48 divided by 3. So that would be 16, 16, and 16. Or if I do have a weighting factor, let's say this stocking level, so 2 to 2 to 4 or 3 to 3 to 6, that's also 1 to 1 to 2. So in that case, it would give me 12, 12, and 24, so twice as much as the others. And now I'm ready to do my chi-square test again. So it's exactly the same procedure, except in this case we just compare one column here, observed versus predicted. So 8 minus 16 divided by 16, 16 minus 16 divided by 16, and 24 minus 16 squared divided by 16. So that would give me a chi-square value of 8. So that's for my 1 to 1 to 1 expectation. And if I do the same with this column here for my 1 to 1 to 2 expectation, my chi-square value will be 2.6. And so the implementation in R is just as straightforward. So in this case, my contingency table is a simple string. So just my single counts for each class, so 8, 16, and 24. So those are these three numbers. And you just run a chi-square test and it will automatically do a goodness of fit test instead of a test of independence. So in this case, I'm asking how well does my observed data fit my expectations. So that's why it's a goodness of fit. And in this case, where I do the test of independence, the reason why it's called test of independence is that I'm testing whether survival is independent of species. So the null hypothesis is that species has nothing to do with survival. And this alternative version here, goodness of fit test, this can be used for any kind of expectations. So it's, for example, used also in genetics where you have homozygote and heterozygote genotypes. So let's say an AA, an AB, and a BB. And you always have a specific expectation of the proportion of those. This is just basic Mendelian genetics. And if that's broken, so for example, if I have very few heterozygotes below expectations, that means that there is some inbreeding going on in that population. So those goodness of fit tests are actually quite widely applicable in environmental and life sciences. So it's good to know how they both work. And this first version here is basically you test the expectation that they're all the same. If you do have an expectation like we had above, like the 1 to 1 to 2 ratio, then you can state this here. So in this case, we have 1 plus 1 plus 2 is 4. So 1 quarter, and 2 quarters is 1/2. Or the other way that you can state your expectation is you just put your weighting factor here. So if your weighting factor is 3 hours, 3 hours, and 6 hours of trying to catch each of those species, then you can put that there. And then you have to say rescale p equals true. So then it converts this p into proportions that add up to exactly 1. And last but not least, we cover the z-test for proportions. So that's actually a much better one than Fischer's exact test or the chi-square. So what you're doing here is essentially a t-test on binomial data. And you can do this by recoding this into ones and zeros. So my survivors are getting a code of 1, and the ones that died get a 0, or the other way around, it doesn't really matter. But if I have these numbers for a species, I can now calculate a mean and a variance. So the mean is, in this case, 1 plus 1 plus 0 divided by 3. So this would be 0.666. And I can also calculate a variance. So the variance is 0.6 times 1 minus 0.6. If they were all ones or all zeros, then my variance would be 0 because they're all the same, right? So this formula does this for you. So if one of them is 0, the whole term becomes 0. And once I have a variance, I can also calculate a standard error. So this is exactly the same idea. So it's the variance divided by n and then the square root taken. And with that, I'm ready to do anything you can do with t-tests. You can now also do with proportions. So that's really awesome. And they have the same statistical power, so it's an order of magnitude better than the chi-square tests. So just like in the single sample t-test, we can pick a single sample and ask, does that beat a particular value? Let's say, does species B have more than 50% survival? Maybe in a reclamation experiment, this is the target. So if the species doesn't have more than 50% survival, then it's just unsuitable. It's not cost effective. So we can come back to applied questions just as we had it before. So your boss tells you have to find species that beat at least 50% survival. Otherwise, we can't afford it. And you can answer those questions. How sure are you to beat the 50%? So extremely useful tests, and it works exactly like the t-test as well. You have your signal to noise ratio. For the species B, my survival was 0.08, so 16 over the total of 20, and so 0.8 minus 0.5, that's my signal, and my noise is the standard error here, and that actually needs to be calculated from my reference value, so from my 0.5. So in that case, it would be 0.5 squared divided by 20. And if you work that out, you come up with a z-score of 2.68. And you can just use the p-norm function that we had, 1 minus p-norm. So that would give you a one-sided single sample test result. P-norm of 2.68 is 0.0036. So you can say that with 99.6% certainty, you can assure your boss that this species will beat the 50% survival threshold. Now, unlike the t-test, this test does not have a adjustment for small sample sizes. So you have to have the central limit theorem kick in for your repeated sample to actually come up with a standard error that's normally distributed. So if your sample sizes are too small, this one will not work too well, and it becomes inaccurate. And the rule of thumb to get around this is that your reference threshold has to meet the condition that the sample size times the reference threshold is larger than 5, and the sample size times 1 minus the reference threshold also has to be larger than 5. So the closer you get to either 1 or 0, the more problematic it is, because your standard errors become too asymmetrical if the sample size is not large enough, because they bump against the 1 and the 0 on both sides. But with this condition fulfilled, your p-values will be reliable enough. And just like for the t-test, there's also a two-sample version here. So if you want to compare species A and species B, rather than species B against a particular threshold, so you calculate the other proportion here. So 8 over 20 is 0.4. So survival of species A is 40%. And then just like in the t-test, it's the difference between the pooled standard error. So the difference is 0.8 minus 0.4. And my pooled standard error is just based on the average frequency. So if you have 0.8 and 0.4, like in this example, your average frequency would be 0.6. But since those can have different sample sizes, you actually do the same procedure as for the t-test. So you calculate a weighted average here, and the math is very similar. You can see if you can work that out yourself. So again, you can use the p-norm function of 2.58, so that's my z-score, and you get a p-value of 0.0049 for a one-sided question. So if you're asking, is survival of species A larger than species B? If you're asking whether it's different, you just have to double that. And again, you should do your check for minimum sample sizes. So in this case, it's actually a fail. So you have to test each of those. And for the 0.8 one, for species B, the n times 1 minus p fails, because that's 20 times 0.2, and that only gives you 4. So in that case, that p-value is probably not 100% reliable. But these are super useful tests. You can get confidence intervals, so they are given. They are asymmetric, they are asymptotic, so they run against the 1 and the 0. But everything works nevertheless, so you can use this just like you would use t-tests and pairwise t-tests following up on an analysis of variance. So in this case, you use them to follow up on a chi-square test, and you can do everything that you can do with parametric statistics. So it's a super useful tool for you to have under your belt. And with that, I'll let you do lab 11, and you can try all these things out.